Have you ever played the game Telephone? One person whispers a message to another person, who repeats it to yet another person, and so on. By the time it reaches the final person, the message has changed. In a similar way, DNA changes slightly each time it is passed from generation to generation. This is Chapter 18, Section 3, Molecular Clocks. Key concept. Molecular clocks provide clues to evolutionary history. Main ideas. Molecular clocks use mutations to estimate evolutionary time. And mitochondrial DNA and ribosomal RNA provide two types of molecular clocks. First, main idea. Molecular clocks use mutations to estimate evolutionary time. In the early 1960s, biochemists Linus Pauling and Emil Zuckerklant proposed a new way to measure evolutionary time. They compared the amino acid sequences of hemoglobin from a wide range of species. Their findings show that the more distantly related two species are, the more amino acid differences there are in their hemoglobin. Using these data, they were able to calculate a mutation rate for part of the hemoglobin protein. Molecular clocks are models that use mutation rates to measure evolutionary time. Recall that mutations are nucleotide substitutions in DNA, some of which cause amino acid substitutions in proteins. These scientists found that mutations tend to add up at a constant rate for a group of related species. As is shown on the screen here, the rate of mutations is the ticking that powers a molecular clock. The more time that has passed since two species have diverged from a common ancestor, the more mutations that will have built up in each lineage, and the greater the differences between the two species at the molecular level. To estimate mutation rates, scientists must find links between molecular data and real time. Often, this link comes from the timing of a geologic event that is known to have separated the species they are studying. If scientists know when the species began to diverge from a common ancestor, they can find the mutation rate for the molecule they are studying. For example, scientists know that marsupials of Australia and those of South America diverged about 200 million years ago, when those two continents split. A link can also come from fossil evidence. Scientists compared their molecular data with the first appearance of each type of organism in the fossil record. Using these dates, they confirmed that the number of amino acid differences increases with the evolutionary time between each group of species. The number of amino acid differences between human hemoglobin and the hemoglobin of several other types of organisms is shown on the screen here. Human hemoglobin is most different from species that diverged earliest in evolutionary time. Second main idea. Mitochondrial DNA and ribosomal RNA provide two types of molecular clocks. Different molecules have different mutation rates. For example, some sequences of DNA accumulate mutations relatively quickly in a lineage, while others have very low mutation rates. Depending on how closely two species are related, scientists choose a molecule with an appropriate mutation rate to use as a molecular clock. Mitochondrial DNA is DNA found only in mitochondria, the energy factories of cells. The mutation rate of mitochondrial DNA is about 10 times faster than that of nuclear DNA, which makes mitochondrial DNA a good molecular clock for closely related species. Furthermore, as is shown on the screen here, mitochondrial DNA is always inherited from the mother, because the mitochondria in a sperm cell are lost after fertilization. This type of inheritance is different from that of nuclear DNA, which is a combination of DNA from both parents. Scientists use the fact that mitochondrial DNA is passed down unshuffled to trace mutations back through many generations in a single species. In fact, mutations in mitochondrial DNA have been used to study the migration routes of humans over the past 200,000 years. Ribosomes, the organelles that manufacture proteins in cells, contain ribosomal RNA. Ribosomal RNA is useful for studying distantly related species, such as species that are in different kingdoms or phyla. When studying the relationships among species over longer timescales, it is best to use a molecule that has a lower mutation rate. Ribosomal RNA has conservative regions that accumulate mutations at a low rate relative to most DNA. Over long periods of geologic time, mutations that do build up in the ribosomal RNA are of different lineages and are relatively clear and can be compared. American microbiologist Kyle Wois first used ribosomal RNA to establish that archaea diverged from the common ancestor they share with bacteria almost 4 billion years ago. As you will learn in the next section, these findings supported a reconstructing of the tree of life at its highest level. This video has been Chapter 18, Section 3, Molecular Clocks. Key concept: molecular clocks provide clues to evolutionary history.